Good evening, everyone, participants, guests, and Professor T. Rao. I once again welcome everyone to this live session in this 3 weeks national workshop on statistical analysis of biological data. For this session, we have Professor T.R. Rao with us, and we already have privilege to hear from Professor Rao in our inaugural session where he gave an interesting and engaging talk on Introduction to Biostatistics. Professor T.R. Rao is currently visiting professor, Indian Institute of Science, Education and Research, that is IISER, Berhampur-Odisha. He has done his Ph.D. from University of California, USA, and he has held various positions before his current assignment.
There was some.
Yes, sir.
All this introduction has been done on 17th. So why do you have to repeat? They all know me by now.
Maybe some students, some participants may have missed it. So
Yeah,
just
go ahead.
to,
Okay,
yes,
sure.
yeah. It will be very brief because I will not be taking much time.
Yeah,
So
yes.
he has, okay, thank you, sir. He has held various positions before his current assignment. Earlier, he was professor in Department of Zoology, University of Delhi, Director, School of Environmental Studies, University of Delhi. His research interest is in ecology and behavior of zooplanktons and fish. And his teaching interest lies in biostatistics and ecology, along with other diverse topics. He is an adorned scientist with many distinctions like INSA Teacher of the Year Award 2015, Canadian Government Postdoctoral Fellowship, just to name a few. I would not like to take much time, and by this, I would like to invite Professor Rao for giving his talk on normal distribution pattern and binomial distribution. Just for the participants, it is an interactive session, live and interactive session. So questions, your queries are welcome. You can post your questions during the session if anything arises in your minds. So you can post it in the chat in the Teams, or you can post in the Telegram. There are 2 sessions for today, and we will be taking the question and answers for both the session after the session 2. So over to you, sir. Thank you.
Thank you, Tabasum.
Thank you, sir.
First of all, I would like to thank the whole organizers of this workshop, in particular, Dr. Wattle, for what I call as spreading literacy. And this is the kind of literacy comparable to what the government was doing, spreading literacy on how to make people read and write. So the kind of literacy campaign you have undertaken through these workshops is statistical literacy, okay, which scientists do need, all right. But a lot of people don't realize that everybody needs this statistical sense of looking at things, in even in their day-to-day lives. So with that kind of a objective in mind, the mission you have undertaken is really praiseworthy, and all my appreciation to you for spreading this. And I really hope that this workshop really helps to inculcate that kind of statistical sense in all these participants who are going to be our next generation of research biologists. Okay. So thank you so much. And also thank you for giving me this opportunity to participate in this wonderful workshop. And it is my pleasure to do so. All right. So, my dear students, I see about 45, around 45, 50. You have heard me on 17th. I will repeat what I said. I am not a statistician. Okay. So I don't know that makes you relax or get disappointed. I don't know. But basically, I'm a biologist, an ecologist. And I have developed what I call as a zunoon, if you want to say, or a passion for this statistics, because I find I can test a lot of my hypothesis in a sensible way and reach plausible conclusions only by applying statistics. So, with this mind in mind, I have been teaching biostatistics for biology students for the last 25, 30 years or more, and I have developed some my own way of teaching this subject. In this last 13 days, you might have learned what I'm going to talk about, but very often the organizers find they have their own constraints in arranging the sequence of topics because it all depends upon the type of speakers availability and their dates and schedules they give. So, with that introduction, now I will start my presentation. Okay. Now let's see. Does everybody see the screen
Yes, sir.
and hear my voice well enough?
Absolutely clear.
Wonderful. Okay, so here we go. So we're going to discuss normal probability distribution. Our statistical distribution patterns will have this word probability because we are talking about the probability of those distributions, not certainty about these distributions. One of the most common distribution patterns is normal distribution. I don't know why it's called a normal distribution, but there is nothing abnormal about other distributions. Okay, this is also called Gaussian distribution after the famous mathematician Gauss. Okay, it is attributed to him, although I'm not too sure whether actually he is the one who discovered this uh, distribution. Typically, it looks like a bell-shaped curve. So everybody knows when you talk about bell-shaped curve, they probably are referring to normal probability distribution. The other two distributions which we'll be talking about in the next session are Poisson distribution and binomial distribution. These three are the most commonly found in statistical applications in biology, of which normal probability distribution by far the most common in biological, social, psychology, and many other subjects. This is the most common probability distribution and hence widely applied. And it's one of the most important ones in the sense that it gives you some possibility of making some predictions, assuming that the parameter you're studying is normally distributed. Now let us look at some aspects of this normal distribution. Some common examples, your own heart rate. Okay. In fact, this is one quick, anybody can do exercise in the class. If you have a class of 20 students, ask them to take a pulse rate for a minute and record on the blackboard and plot the frequency, and presto, you have a more or less normal distribution, a bell-shaped curve. Don't bother about some deviations from this normal curve at this stage. We'll explain those later. So you can see heart rate normally in adult humans at 72, we know that mean value follows more or less normal distribution. It doesn't have to be always. I'm sure those who are of you who are listening, if we have one of those watches that gives you heartbeat, you can see that doesn't have to be. For instance, mine probably says, let's see, something like It is showing 95. Oh, wow. Okay, so this is a normally distributed parameter. Intelligent quotient is a normally distributed parameter. World over, they say that the mean intelligent quotient value is 100. So it hovers around this. And if you have the extremes here, it could be an Einstein, something like this. That's also a normally distributed parameter. The distribution of marks in my course at IISER Berhampur also follows a normal distribution, more or less, of course. See, this is the distribution of marks, and this is the frequency I plotted. It gives me a fairly decent normal distribution. Even pizza delivery times in large cities, lo and behold, follows a normal distribution. How about that? So most of the cases, the normal average time for a guy to deliver pizza at your door after you place the order is about 45 minutes. You can test it out in your city. So I used to do a simple practical exercise with my students. For example, beans, Mexican beans. We ask this question: is the weight of Mexican beans a normally distributed variable? Okay, we'll see. So we have asked the students to take beans, maybe 10 or 15 for each person, student, and they go and weigh on a sensitive balance to the nearest tenth of a or hundredth of a gram. So here are the values. Okay. And you take the frequencies, okay, and use these ranges, 81 to 120, 120 to 160, etc. Okay. The way you choose these things is that you should get some bars, frequency bars, around 10, 8 to 10, in order to get a decent looking normal distribution pattern. So that's, below and behold, we use these 220 to plot a frequency, and it shows a fairly good normal distribution. So you can see that many of these parameters, they follow a normal distribution. The larger the sample size, the greater the approximation to normal distribution. If you plot the cumulative frequency, okay, as the table shows for the same data, Mexican beans, it shows a typical S-shaped sigmoid curve. So if your data show cumulative frequency as sigmoid curve, you can be fairly confident that is a normally distributed parameter. So let us see what are the attributes of normal distribution. That is the central position of where most of these values are located. Okay. So if you are looking at the heart rate of say 200 students, most of the values must be hovering around this. That is A. And the spread is this B you see on both sides. Some of them are lower or much lower than the normal heart rate, and some have higher heart rates, and some extremes, as if you're doing a treadmill exercise. And that's spread in common man's terms, but in statistical terms, we call dispersion. So we have to know how to take care of these two parameters in order for us to quantitatively describe normal distribution. So let us first look at measures of location. There are three methods: mean, mode, median. Within mean, there are three measures: arithmetic mean, which is simply this formula, and this is the most common one used. When people talk about mean or average, they are talking about arithmetic mean. But there are some special situations, some special kind of data that would require you to look at geometric mean, which is actually using the log values. Okay. In certain situations, in a peculiar kind of data, you might require harmonic mean, which is actually taking the reciprocal of the values that you have collected and use this formula, you develop a harmonic mean value. So at this stage, I will say that you be content with arithmetic mean because it's most of the time people use this only. Depending upon the type of research and the data you collect, you might require either this geometric mean or harmonic mean. Mode is simply the value at which most number of values are located. Okay. Now, median is halfway point in the set of observations which are arranged in a descending or ascending order. The difference between mean and median can be illustrated with this example. So, if you look at this, in order for you to balance it, you have to hold it there. I'm sure you know. So, that actually is the mean. Okay, it distributes the total amount equally. Whereas median is half the length in the spoon. That means half the observations in a normally distributed variable will be below this point and half above. Okay. So you can see how if you put bar diagram, this is how they would look, the difference between the mean and the median. Now I pose some questions now, not questions, statements. I pause and you, dear participants, think what could be the common statistical term when we talk about this, things like this. Is it mode, median, or a mean? You don't have to put the answer in the chat box, okay? The class average in biology was 52. What statistical term are we referring to here? Half the village population are below poverty line. Here, here. Okay. So here most students use, majority of the college girls, these two refer to the mode. Class average refers to the mean. These two refer to the median because we are talking about half the population. And here 50% of the fruits ripen by the 5th day and 50% don't. Right? If you look at the, in terms of percentages, median is after all a 50% level, right? So we call them percentiles. And there is no reason why we only stick to a median, which is 50 percentile. You can choose any percentiles. So the median at 50 percentile, you can choose 50 percentile or 25%. It's called first quartile because it divides the whole set of data into 4 pieces. Sorry. This is called the third quartile, will be 75%. So, and you can have any percentile. This range between first quartile and third quartile is called interquartile range. Okay, that's the formula here for you. Now, if I use a 5% there, 5 percentile, okay, very often you see that in SAT exams or GRE exams, they say you are 5 percentile or 2 percentile and all those things. What exactly does this mean? It simply means in the exam taken of all the 10,000 or number who have taken that exam, you are in the top 5%. Okay. So if the university says we'll give admission to only those who got 5 within that 5 percentile, that means all these candidates who got above 5%, I mean this percentile marks, they are eligible for admission. Not that they will all get admission. universities like Harvard might simply say, we'll take only the top 1%. This depends on the elite institutions. So you can see we arrange, but remember, you have to first arrange the data in ascending or descending order. Generally, people talk about a 5-number summary for a set of data. You give the minimum value, first quartile value, the median value, third quartile value, and maximum value. And very often we speak of outliers. Those are actually any observation which is more than this value. So generally outliers are looked upon suspiciously in the sense, did I measure right properly? How come it is so far away? So those are the questions that arise when you see a number that is far from what you would have expected. Something like you take the pulse rate and he shows 32 or 250. There must be outliers. If it goes to 30, 32, he would be in the hospital or on a ventilator probably. Okay. Now I'll just give you an example. Somebody has measured spider silk, how strong it is. As you probably heard, spider silk is for its weight, it's one of the strongest silks known. So they took measurements just to show you. They took these mean, median, and all those things now. That's a median, first quartile, third quartile, and the minimum value and the maximum value. So these are the 5-number summary. Now you can also look at, see, in this array of data that you have, do you have any outliers? Use the formula I have shown you in the earlier slide. You will see that 479 and 740 are outliers. Okay. But very often the question asked, for our consideration, do we use mean or median? That's an important question, because in a lot of say social sciences, psychology, linguistics, all those things, they are called non-parametric statistics. Somebody must have given a lecture, or will be giving a lecture on non-parametric statistics in this workshop. But basically, most non-parametric statistics tend to use median as a measure rather than mean. Okay, so you have to know whether you want to use mean or median for your statistical analysis. So there are some situations in which you will use mean or median. Now, so we looked at the central location of the points. We look at the dispersion, the amount of spread. Now you can see the spread is more in diagram B than in A. One way of looking at it is the range, the largest value and the smallest value. The other one is a statistical measure called standard deviation. That is the formula, and I'm sure you will find in textbooks. I'm not here to conduct an exercise for you to calculate standard deviations. So you see mean and standard deviations. Here the means differ, but standard deviation is more or less the same in A and B curves. The spread is approximately the same, only the means differ. Now here, the means are the same, but the standard deviations are different. The dispersion of A is much more. Here, both mean and deviation also differ, A and B, right? So very often when you are looking at a few characters, suppose you are looking at a flower, some variation, petal size, overall tube size, or anther size, stamen size, all kinds of things, variables that you can measure in a flower for whatever study you are doing. You want to ask sometimes what character is better I choose, okay? Normally, the statistician would advise you take a character that is less variable because it should not by itself show a lot of variability. So it confused whether the variability is inherent in the population or caused by some environmental factor. Okay. So in order to do that, we use a measure called coefficient of variation, which is simply um, the standard deviation divided by the mean times 100, so that coefficient of variation is always expressed as a percentage. So you would like to choose a variable that has the least amount of coefficient of variation. Often that authors quote when they say this character shows a lot of variation. For instance, the annual temperatures in this region show a lot of variation. In brackets, they will choose to say CB is equal to 25% or something like that. Okay, so it's commonly done to indicate whether the parameter that you're studying is variable or not. So far we talked about a sample values which we plotted and it showed some normal distribution, but very strongly you have to remember that the sample is only an approximation to the population at large. Whatever you are trying to do and make a statement about the population, you are not able to do because there is no way you can measure the entire population parameter. Just imagine measuring the wing lengths of all the mosquitoes in your town. Ridiculous even to make a statement like that. We take a random sample and try to see if our parameters that we have measured are good enough to say that they faithfully represent what is happening in the entire population. So our population mean estimates the population mean, which is symbol mu is used. Okay. Our variance, which is simply standard deviation squared, that is our variance that we got in our sample, and we hope that it approximates or estimates the same parameter value of the population. That is called sigma square. And if you remove the square, we have S, which is called standard deviation, which is an estimate of the population parameter of sigma. Okay. So in any study, you're only taking a sample, you're only getting these values. But you want to talk about these values. Because ultimately what matters is not your sample, because you have taken sample in Zakir Hussain College, somebody has taken in New York, somebody has taken in Finland. But you want to make a statement about the entire population. So you have to see how to extrapolate from your sample to the population. The normal probability distribution can be algebraically explained by this formidable looking function. Okay. Mercifully for us, we don't have to use this formula for any reason. Unless you are mathematically crazy, no, I want to test this, etc. You don't have to. But you can see that it uses these two parameters, the mu, which is population mean, and the standard deviation, which is sigma. What is this used for? Look at this now, perfectly symmetrical now. You see, one of these, if a distribution is perfectly normal, the curve also should be perfectly symmetrical. Okay, what do we mean by symmetrical? here is, if you cut this curve, so let us say it's a piece of cardboard shaped in a normal distributed bell-shaped curve. You cut it in half exactly at the mean, obviously. Okay. Okay, two pieces. You reverse this piece, one piece. Okay. Call K. Now see, exact fit. So that's what I meant by a perfectly symmetrical normal curve. That is not to say that many of the observations we take in biology, sociology, anywhere are perfectly symmetrical. No. We are only trying to use this idea that they are symmetrical for various statistical analysis. So when you talk about the bell-shaped curve, the total area within that, all the probabilities of the frequencies, if you add them up, it should add up to 1, obviously, right? So let us see how much of this area is covered by a certain amount of the mean and standard deviation. I want you to follow this very carefully because this has a lot of applications and hypothesis testing also later. So what I did is I put this mean value and indicated the standard deviation by this width. It is 1 standard deviation, 2 standard deviations, 3 standard deviations, etc. It is on the negative because it's going on the left side. Now you see that when I put 3 standard deviations on each side, the curve is more or less taken care. The entire area is taken. A small amount is left here, small amount is left here. Okay. That might give you the impression, which is not right, of course, that if you go to 4 standard deviations on each side, it will cover the entire area. I will say it almost will cover, but never entirely. We'll see how that is. Okay. So now using that huge algebraic function that I have shown you, one could calculate how much area will be covered between mean plus or minus 1 standard deviation. And it can be shown that it covers 68% almost of the area. In practical terms, this means if you made 100 observations on any parameter, okay, 68 of them will lie within 1 standard deviation each side of the mean. Okay. What about 2 standard deviations on each side? 2 standard deviations on each side will cover 95% of all the values. Okay. So this means now in your 100 observations, 95 should lie in this pink area. Now let's extend this further and see what about 3 standard deviations. Lo and behold, 3 standard deviations will take care of 99.7 of these observations. You know what this means in terms of taking out the decimal and making a statement? If you made 1,000 observations, okay, 997 will lie in this area, which is defined by mean plus or minus 3 standard deviations. That means out of 1,000, 3 still will be outside this range. As I was telling you before, if you go to 4 standard deviations on each side, this will not become 100. That means it might be 99.7 or 99.85, 99.97, whatever, but you cannot say it will reach 100. You know why? These extensions we call as tails, the tails never touch either 0 on this side or 100 on that side. That's the property of the normal distribution. You can't challenge that. Okay. This is very important for you to understand. Okay. Now, that was easy to do because we know we are defining in terms of one standard deviation or two standard deviations on each side of the mean. But what if we have an observation that says mean plus 0.7 standard deviations or 1.3 standard deviations? Then you have to have a way of calculating that particular area. So now we have this status, right? I told you what it means if you extend it to population. Now I ask you, not one standard deviation on each side, I will say 1.2 standard deviations on each. How much area is covered? If you are crazy enough, you can use that algebraic formula and calculate. But mercifully for us biologists, somebody calculated and made a statistical table that is called the areas under the normal curve. Okay, I'll come to that in a minute, the table. So now this way I presented here, that's called standard normal distribution. A standard normal distribution is one in which the mu or mean is 0 and standard deviation is 1. That becomes easy for calculations and also for making tables, area tables. That's standard normal distribution. Okay. So we use that formula and develop this whole table. It's really available. Your organizers will be able to give you a link for this or send you one. I don't know. Okay. So you can see that. Suppose now we wanted to see for 1.2 standard deviations. I can see that is 0.3849. Okay. Now, this values is only for the 50% of the curve. You know why? Because it's exactly the same on the other side, except with the negative sign. So the unnecessarily they don't want to show for the entire curve. They are showing for the one half. You can, since there are no negative signs here, I would say this is the right side. So now you can see that for 1.2 standard deviations, it will be 38%, 39, almost 39%. So this is what I meant. They are showing only this side. But the other side, one, if you say 1.2 standard deviations on the left side, is simply this is the same value but with a negative sign. If you take the given value, subtract from the mean, and divide by the standard deviation, that's an important measure. That is called standard normal deviate, or also called Z. A given value minus or plus, I mean, because you can do both ways. The mean, if this is not the population mean, it will be your sample mean, y bar, and standard deviation, yes. That is called a standard normal deviate. Z is an extensively used parameter for various things. One of them is in assigning grades in courses. This is what I do. I don't go by the absolute marks. Okay, in giving grades A, B, C, D. I plot this and see in terms of Z values or standard deviations. All these are one standard deviation, two standard deviations, etc. So now I can say grades. I might say all these students will get A, two standard deviations and above. These guys get B, C, D, and there are one or two marginal cases. I might be tempted to give an F here. But it just depends. If the student did well in other things like internal assessments, etc., maybe I can just push him above that line. So this is normally done. And this is the most objective because most often, depending upon the toughness of the test, absolute marks do not make sense. Okay. So we come to the what I mentioned, that a perfectly distributed, normally distributed variable allows you to make some predictions. Okay. And since we know that many parameters are normally distributed, we can use this to make some predictions. So here is one. What percentage of the population expected to be taller than 185 centimeters? I'm giving you enough time for you to guess. Okay? Okay. So now you see which area is that now, so that you can immediately figure it out. And this percentage, this much of the area, they are above 185. So taller than 185 centimeters. So whatever this percentage is, that percentage of the values people will be taller than 185 centimeters. Okay. How much is that? Okay, before I tell you the answer, this we have seen that two standard deviations on each side will cover 95% of the values. So what is left is 5%. Okay, actually 4.5%. So but it's equally distributed on both sides. So this will be 2.25 and this will be 2.25. That means if you measure the heights of 100 people with this population mean, sample mean and standard deviation, you can expect only about 2 in the sample individuals will be taller than 185 centimeters, only 2 out of 100. And 2 will be shorter than 155. Okay, that's how you do. Now, here is a situation where it's not exactly falling on a 0 or 1, 2 like this, but in decimal value. See that? Now, I'm asking this, sorry. The weight range of 10.2 to 15 milligrams of the beans, fruit flies, sorry. Okay, so this is the weight range. Okay, 15.5 to 10.2. This is the area, the orange colored one is the area that covers it. Now we want to calculate this area so that we can express in terms of percentage. You can calculate the Z, which means these values minus the mean divided by standard deviation. It tells you how many Z values it is away. Then you can look up the table. Okay, you remember that the normal curve area table, you look at the Z value, it will tell you how much area is covered in this one, and that corresponds to the percentage of the population which is expected to be in the weight range of 10.2 to 15 milligrams. Okay, that's how this is giving us the ability to predict. You might ask all kinds of questions. For instance, if you take the weight of newborn infants, just immediately after birth, which is done in the hospitals, they weigh the baby. So if you gather all those statistics, say 1,000, you gain a data set in which you can present with a normal curve, and you see beautifully it forms a normal curve with the larger sample size yield, very symmetrical looking normal curve. From that, you can immediately say how many babies are born underweight that their survival probability might be very low unless they are kept in an incubator. The same way, you can see how many babies are so heavily born at birth that they must have given a lot of trouble for the mother during the delivery time, or perhaps, who knows, they might end up being very obese in their life. We don't know. But we say, of course, very healthy baby, oh, isn't she cute and all that. So this gives us an idea about the way to be able to predict certain things from normal distribution area. Okay. I'm giving the answer. So in case you have been mentally working or quickly working on a piece of paper with your calculator, you can verify that this is the answer. So do you think the normal distribution is always perfect like this? No, no. There is a statistical simple rule of thumb. Any parameter, any thing variable that you talk about in biology, any science, okay, even if it is not normal distributed, if you start collecting enormous number of observations, ultimately they tend to form a normal curve. Okay. Okay. This means now, as you can increase your sample size, things start beginning to look more and more like normal distribution, typical bell-shaped curve. This is also the reason to say that when you have smaller sample sizes, deviations from normal distribution might be so much that you might get a different kind of statistics to deal with those data. And for all practical purposes, although we assume that our data are normally distributed and go ahead and do some what are called parametric statistics, very often it makes sense for you to check whether it's actually true that they are looking normal. But if you have enormous sample size, like I had 220 Mexican bean weights, or you took measurements of IQ from the entire college students, there might be the frequency distribution might just begin to look like a perfect normal distribution, but it's not always so. You can see that there will be deviations. There are two types of deviations from the normal distribution. One is called skewness. The skewness is stretching out in one direction. It could be stretched out in this direction, then it's called left skewed, otherwise it's right skewed. When I show this diagram, you should start thinking what kind of sampling distribution or in my sample would lead to this kind of a curve. Instead of a normal distribution curve, why is this stretched out? You should keep thinking, imagine a situation where you are doing some sampling of some people or some fruit flies or some hibiscus flowers. What are the conditions under which this skewness might appear? The other one is kurtosis. Kurtosis is either too much flattening of this curve, as the line shows, or too many values lumped towards the center and not evenly spread out throughout the curve. Just too many values hovering around the center here. Here, too evenly spread, too much evenly spread without showing any peak in the center. So these type of variations refer to kurtosis depending on which shape, either leptokurtic or platykurtic. So basically, in the two deviations from normality are skewness and kurtosis. And the next question is, so what do you want me to do if my data show the skewness and kurtosis? Well, we say you have to deal with it because any analysis you do by ignoring them might lead you to conclude in a different way and reach wrong conclusions. You have to do something about this too. Either you say my sample size was too small, maybe if I increase the sample size, the skewness and kurtosis will go away. Or did I do something wrong while sampling that I got this messy looking curve? Okay, here is the question now. You see, that is the situation you are in now. Okay, normally you look at the annual income and frequency based on your sampling of these shoppers. Now, pose a question. Suppose in your sample of these people that you have asked, excuse me, if you don't mind telling me your annual income, please. They might get suspicion, say, are you from income tax department or what? Why are you asking me? I don't want to tell you or something. But suppose they know it's only a student exercise, they're just doing some exercise, they're collecting some sample, I'd be glad to tell you what they're. Okay. Suppose now, just assume that inadvertently, without your knowledge, because you've never seen him, in this random sample, you have included any big guys, okay, Ambani, let's say. What would happen to the curve? There will be a steep increase here because you see some 2 or 3 in the sample who are very rich without showing that they are. They might be still standing near the McDonald's or something or Pizza Hut without going to any fancy one. And you randomly happen to sample them, and they are very sweet and nice, and they just told you. And so your curve has become skewed. Same way. So deviations from the other side also can happen. If you are sampling, say, IQ values, okay, of a normal set of people in a colony, and you tend to get about normal curve. But if you suddenly happen to have some few politicians there, and suddenly it might be left skewness, it might be there. Okay, you got it? Okay, so these are the 2 deviations one tends to get in normality. So you can see basically are all put together. This is a normally symmetrical one. Here it is skewed and it's right skewed. Okay. And you must also mentally see what happens when the skewness happens to the positions of the mean and median. Okay. Just watch and see. You can work it out yourself whether the mean will be higher than median when skewness happens or not. Okay, there is another parameter that you need to for further analysis in these statistical tests. The philosophy of that is, let us say from a normal population, you take subsamples, 20, 30, whatever, okay, and you calculate mean for each one of the samples, right? And plot those means here. So these most likely, because this came from a normal distribution, a large population, these subsamples also should show normal distribution and their means will be normally distributed. Okay, that's the statement we can make. This is this is a very simplified version of central limit theorem. A good statistician listening to this will contest and say, no, you can't simplify like this. There is a big logical explanation in algebraic formula, etc. We're not concerned. Basically, what we're concerned here is when the sample size becomes large, number one, it becomes normal distribution. And these means of the subsamples that you have plotted, the standard deviation of those subsamples, that is in this blue curve here, will be what we call as a standard error. Okay. standard error is conventionally calculated in terms of a formula, which is your standard deviation divided by root n. I'm not going to derive why we are using this to get our standard error, but take it from me that if you use standard deviation that you got for your sample and divide by the square root of your sample size, you will get what is called a standard error. And you can see that standard errors is a very important one for building confidence intervals for your mean. But here you can see standard error can increase, okay, if variation is increases, standard deviation increases, or if sample size is smaller. So if you want to keep standard error small, you have to increase the sample size or reduce the variability in your sample. Okay. So the parameters that I have described so far are collectively called descriptive statistics. In all statistical software programs, these are listed as descriptive statistics. After doing this, entering your data and all that, if you go for descriptive statistics, it will give you a whole bunch of a table with all the values like this. This is for the Mexican beans data. So basically, see, you can see that all the parameters that you required, okay? See, my sample size was 220. Based on that, it gave you all the parameters. And it shows the kurtosis and skewness also. Normally, it should be both should be 0, but pretty good. You know, it's 0, 1, almost 0. So skewness is not there. Kurtosis also is not too far away. A little bit of that, but I would say this my sample of 220 gave almost a perfect normal distribution. So that I could have simply asked how many beans in my this 220 would be weighing less than so and so grams. Okay. It gave you the mode, median. This is also a good indication. When the normal distribution of your sample is perfectly normal, that's perfectly symmetrical, the values of mean, median, and mode will all be exactly the same. Okay? When they're exactly same, you know that the distribution curve is very perfectly normal. Here it's almost normal because 0.27, 0.7, 0.276. Tiny difference. So this means on a large sample size of 220, I get a perfectly normal distribution with all means and median and mode being equal, kurtosis and skewness being minimal. Well, finally, we ask this question, why is it important to know if my data show normal distribution or not? So the reason is this. Many of the statistical tests we perform, whether it is a t-test or an f-test and all those things, they are done on normally distributed parameter. Okay, that's why they are called parametric statistical tests. Okay, so when you start doing these f-tests and t-tests, you are asking the reader to assume that my data are normally distributed. Okay. Whether you have actually checked it out and told the reader so, we don't know. But if the referee, the reviewer is picky, he might simply ask you what were the skewness and kurtosis values, etc. So it's very important for you to check whether your data are normally distributed or not. What to do if they're not normal? Well, there are various possible ways. You can abandon conducting parametric tests like t-test and f-test. Statisticians have worked out a non-parametric equivalent of all these tests. They are collectively called non-parametric statistics. The very term non-parametric means They simply don't expect your data to show mean standard deviation like a typical normal curve. Okay, and still you can use some statistical tests on them. And a good statistics department might have a whole one semester course on non-parametric statistics, because that's extensively used in non-biological sciences like sociology, philosophy, psychology, education. So that's a very important non-parametric. So if your data don't show parametric distribution, you simply go to non-parametric. Otherwise, you can also transform your data to see if the transformed data look like normal. We are not asking you to change the data, it's transform them, say from standard arithmetic values, convert them to log values, and if the log values suddenly look normal distribution, fine, do that. Okay, so that's how we deal with the problem if the distribution is not normal. Either you go to abandon parametric statistics, go to non-parametric statistics, or see if you can transform your data in such a way that they turn into parametric values. Okay. Okay, I think I'm getting close to the time, so I'm just giving you some numerical problems that you can try. I don't expect you to try now, but I think somebody recorded this lecture or this slide itself. They can pass it on to you as homework problems or practice problems for you to do later. Okay. So we stop here, and thank you so much.
Thank you so much, sir.
Okay. Yeah.
Yes.
Matt?
Yes, sir. So this is, we have come to end of the first session, and we will take a break of 10 minutes in between before we start our session 2.